Okay, so we have transcribed the DNA into RNA. We have got our RNA strand, we're done, we're ready to go out in the cytoplasm with it, right? Wrong. Not if we are talking about a eukaryotic cell. If we are talking about a eukaryotic cell, we have got to do some tinkering with this stuff. It's not ready for prime time yet. So what we are now going to talk about is RNA processing, which is what has to happen to RNA in a eukaryote. This applies only to eukaryotes before it can go out into the cytoplasm. From the nucleus, of course. Remember, in prokaryotes there is no nucleus, but in eukaryotes there's a nucleus, and more importantly, the RNA has got to be modified before it can be translated. Okay, so RNA processing then is kind of phase 2 of transcription, if you will, occurring only in eukaryotes. We've got to modify the ends of the RNA and the middle, okay? Now, RNA processing used to be called the removal of junk information. That is not the way we think about it anymore. Nature does not make a whole lot of junk. And these things that we're going to remove from this RNA molecule are likely very much not junk, but they also aren't needed perhaps for what the cell is trying to do at this point in time. Let me explain further. Eukaryotic genes have some stuff with some nucleotide sequences within them that are usually not useful to the protein you're trying to make. The part that is relevant to the protein you're trying to make is called the exons, the real part of the gene, the expressed coding RNA that came from good coding DNA. Okay? There's another part in there that is introns, and that is the junk, if you will. It's not junk, but it doesn't have any use for what we want to do right now. It's an in-between nucleotide sequences. It's not going to be part of the final product. So the introns have to come out, and that's the only way I can remember. It's opposite. The introns are going to come out. So in RNA processing, we are going to cut out the part of the DNA message that we do not need for this protein we're about to make. So we're going to splice. We're going to remove the introns. They're going to be cut out, and then the exons will be spliced together. Okay? So if you look here, these bright red are the exons, the pink are the introns. These are coming out, and then the bright red will be knitted together, and you see we have a much shorter piece of RNA as a result. Okay? So, we did this. So I do want to give you a term. The primary transcript is the RNA before it's been edited. It's also called pre-mRNA. After we splice it, we end up with a mature RNA transcript. Okay? So this is showing you the original DNA, has some stuff in it that we don't need for this particular piece of genetic information that we want. So we're going to cut that out, and when it's in the RNA form, we're going to cut out these pieces that we do not need. Actually, I think I've got that exactly backwards. These are the pieces we don't need, these pinky, peachy pieces, and then we're going to keep what we do need. And that will be the mature transcript. These are the guys that figured this out, and that's lovely. Now, there are enzymes, of course, that have to help us do that. They are called SNRPs, and there's really an N in there, but that's disregarded, and they're called SNRPs. Small nuclear RNA, and also some proteins embedded with that. Okay? And what happens with these little structures is that they come together, they bind to the RNA where the exon and the intron meet, and they curve around and they meet each other in a complex way to become kind of a big glob that we call a spliceosome. So SNRPs make spliceosomes, and they slice the DNA the RNA, and paste it together. They cut and paste it just like that. They take out the intron and they glue the two exons together, and that's what SNRPs do. Again, another really important use of RNA, a way that RNA kind of acts like an enzyme to a certain degree. Okay? All right. Now, a really important concept in biology today, and why it is, humans have only about 25,000 genes that we can figure out. Yet we are extraordinarily complex. This is not a whole lot more than a fruit fly, yet we feel more complex than a fruit fly, and people were astonished that this was all humans had. It used to be taught when I first started teaching that humans had 100,000 genes. That's not right. How can it be that we can be so complex and have so few genes? Well, it is thought that it is because of something called alternative splicing. You can have one gene here at the DNA level, but you can cut and paste it in about a zillion different ways. And however you cut and paste it, you get a different protein as a result. Okay? So alternative splicing kind of explains some of the extra complexity that we are able to achieve with our reasonably few 25,000 genes. We slice and dice that genetic information to make any number of different RNA sequences, which gives us a bunch of different proteins as a result. And it is starting to get kind of hard to define what a gene is, because the same place on the DNA, depending on how you slice and dice it, could code for dozens of different proteins. Okay? All right. Now, there's a little bit more post-transcriptional RNA processing we have to do. We have to keep the RNA molecule safe, prevent it from degrading. So we add a cap called a 5' cap. It is a little guanine molecule basically up at one end, at the 5' end of the mRNA. And then there's a bunch of A's, 50 to 250 adenines at the other end, at the 3' end. That's called a poly-A or many-A tail. And the point of these things is to keep the RNA molecule from degrading. Okay? So, the transcriptional unit, what gets transcribed? It's a gene that gets transcribed. Okay? And at the beginning of that gene is the place where it says this is where you start transcribing, the top box, the promoter. So remember, this is DNA we're talking about. And there may be regions in front of that that help enhance its transcription. RNA polymerase would attach to that promoter and it would transcribe that gene. And when it did so, and it got to a stop place, it would stop, and then we'd get an RNA molecule from that. And that would be a pre-mRNA molecule we would get with the introns and the exons both included in that. And so we'd have to process that, take out the introns, and then we'd have a shorter but more accurate gene of what we actually need at this point in time, that mature RNA molecule, which would then go to the ribosome. We put the 5' cap and the poly-A tail on it to protect it as it went out to the ribosome, and we're ready. All right, so next is what we do with that mature mRNA to translate it and to actually make the protein. So, thank you for watching, and I will catch you in the next podcast. Take care.